Okay, so today's a very special day because we have I think one of our most exciting guests that we've had, and it's our first game changer of the year. And it's my pleasure to host my first ever game changer. So it's a very special personal occasion. And today we have Professor John Mattick. And I think he really does meet the definition of a game changer. For those of you who aren't familiar with the latest on John, he's a Sharp Professor of RNA Biology at the University of UNSW. Before taking up his current role, he was the executive genomics, the director of Genomics England, and he's also with the inaugural director of IMB, director at Garvan, and really he's had so many major positions and accolades. He's internationally renowned for his work in deciphering the role of regulatory RNA in the evolution and development of complex organisms, which today sees him ranked globally as #1 in the field of non-coding RNA. He's a fellow of the Australian Academy of Science, the Australian Academy of Health and Medical Science, and the Australian Academy of Technology and Engineering, which is quite extraordinary. He's also an associate member of EMBO. He's published more than 300 research articles and reviews and more than 100 citations. His accolades include the Gutenberg Chair, the International Union of Biochemistry and Molecular Biology Medal, the Human Genome Organization Chen Medal, and the MD Anderson Bertner Award for distinguished contributions to cancer research. So please, a special hand for John Mattick. Thank you.
Thank you, Maha. I guess if you hang around long enough, you accumulate stuff. It's a real thrill to be here. It's been years since I've been to TRI, and it's fantastic. And it's also an honour to be asked to be giving a game changer seminar. And I hope I do justice to the ambition of the program. I've got a lot of affection for Brisbane and UQ in particular, and it's lovely to be back and to see so many familiar faces and old friends in the audience. Thanks. So it may not surprise you that I'll be provocative today, at least at times. And I want to try and convince you, probably don't have to anymore, but that we've fundamentally misunderstood the structure of genetic programming in complex organisms and the information content of the human genome. Because of the assumption based on bacterial genetics that genes encode proteins, and apart from the infrastructural RNAs involved in that process, that all genetic information is transacted by proteins. So you see the title of the paper, and many of you will be familiar with Thomas Kuhn's famous 1960, we do acknowledge the traditional owners of the land and hope they continue to prosper. So, there we go. Right, so some of you may not be familiar because this is a long time ago, but it was a very influential book at the time. Thomas Kuhn, a sort of scientific philosopher, wrote this book in 1962 called The Structure of Scientific Revolutions. And in it, he posits that science is sort of, and using my terms now, kind of lurches from paradigm to paradigm. That you have a conceptual framework which is generally accepted in your field of science, and that research investigations are done and the data interpreted within that conceptual framework, what he calls normal science. Normal science is sort of turning the handle within a conceptual framework. And that conceptual framework persists until two things happen. The first is that anomalies accrue that don't quite fit somehow. They could be, completely contradictory, but more often than not, they just don't fit. And by the way, I don't think we've got time to talk about the the universe and Big Bang Theory, but that's another one that's got some anomalies, including dark matter and dark energies you might want to think about if you're in physics. But back to this story, anomalies accrue. But the conceptual framework is held on to in the face of these anomalies, and often they swept under the carpet until a second thing occurs, and that is that there is a replacement conceptual framework, which more or less explains the anomalies as well as the pre-existing understanding. In other words, in our minds, we don't move to a different conceptual framework until we got one to move to, despite the fact that there are maybe problems with the existing understanding. So the paradigm that's prevailed in molecular biology really since the 50s and 60s is that genes are generally synonymous with proteins and include most genetic information, including regulatory information, that's emphasized for a reason, is transacted by proteins. And what this basically assumes is that the mechanisms that control bacterial physiology are going to be sufficient to account for multicellular development. As I explained, putting 40 trillion cells in the right places will be controlled, regulated by the same mechanisms that turn the lac operon on and off in response to nutrient availability. That's a big leap, but underpins the assumption that, with genes, proteins do all the stuff. I want to pay some homage to this lady. Some of you will recognize. This is Barbara McClintock as a PhD student in front of her beloved Mays back at the University of Missouri. And she intuited, even before the double helix, that the biochemists were taking things off track. Now, she's a geneticist, and one of my aphorisms, by the way, if you forgive me being philosophical, is that genetics discovers and biochemistry explains. So what she wrote to a good friend, Marcus Rhodes, in 1950 was this: are we letting a philosophy of the gene, that is, as a protein coding entity, control our reasoning? What then is the philosophy, and is it valid? When one starts to question the reasoning, she means the evidence and logic behind the origin of the present notion of the gene, the opportunity for questioning its validity becomes apparent. Now, at this period in molecular biology history, people were doing studies mainly in bacteria, neurospora, microbial genetics, mainly in metabolic pathways. The one gene, one enzyme statement was made, which morphed into one gene, one protein. But despite the fact that she got the Nobel Prize in the 1980s for discovering transposition, she went to her grave frustrated because she said, it's not transposition, stupid, it's control. And what she was observing under the microscope in the cytogenetics and in genetics generally was that there were bits of DNA that were moving around, changing the expression patterns and the phenotype, in this case of the maize. And she intuited that these weren't enzymes. These were control packets. And she then reasoned that there was another level of genetic information which was controlling patterns of expression of the components, I call them the Lego parts, proteins. So back to this. So this is deeply imbued. And I'd say for many years, it was almost impossible to challenge it. And the protein-centric view that's pervaded molecular biology since its inception is really a reflection on the mechanical zeitgeist, I love that word, prevailing sort of cultural paradigm of the age in which it came out of, the 50s and 60s, which was very mechanical. Bicycles were made of parts, and so were humans. There was a part to carry oxygen, this and so forth. And it has led to many subsidiary assumptions, including stupid ones about the nature of enhancers that I'll get to. Despite a number of subsequent surprises or anomalies, that should have given pause for thought. And where I got interested in this was as a postdoc back in Texas in the 1970s, late 1970s. Biggest surprise without question in the history of molecular biology was that genes in pieces. Nobody in their right minds expected that genes in the higher organisms wouldn't be collinear with their protein products. They're mosaics. And in fact, the protein coding sequences of these genes, including things like hemoglobin, were tiny bits separated by huge tracts of something that was transcribed but not translated. But, it was quickly worked out that this was spliced out. Oh, thank God, all's right with the world. Messenger RNAs are still made and exported to the cytoplasm. And the essential attitude, which was universal all the way to Crick, was, and I'm paraphrasing, we don't know what this stuff does, doesn't fit our ideas, so it must be junk. Now, can you imagine? Is that science? Is that curiosity? Is that imagination? So here I am, a little old postdoc in Texas, thinking, after a couple of beers, as I explained earlier today, thinking, hang on, there's another possibility here. This stuff, RNA is doing something. There's something going on here that we did not expect from studying bacterial genetics. So that was a universal assumption. I mean, and this stuff was rationalized then as the hangovers of early evolution of the assembly of genes. And Wally Gilbert is quite famous, wrote about this, but think about how idiotic that statement is. Because what they posited was that these early genes were in pieces, but then bacteria, microorganisms, had evolved these out under pressure for rapid replication, efficient, replication. But our ancestors were bacterial archaea for 2 billion years, and then single-cell eukaryotes for another 1 billion years in the same competitive environment. So what were they doing? Hanging on to their introns, waiting for the sunny day when they became complex organisms and they could carry around a load of junk? I mean, the lack of thoughtful reasoning was just incredible. Anyway, that's probably enough of my rant. I'll get on with actually telling the story now. So it occurred to me that the equally and really more plausible, certainly far more interesting alternative is that other information is being transacted by the RNAs rather than simply through proteins. The second surprise, which actually is slightly earlier, was that our genomes are full of transposon-derived sequences, and on the same dodgy logic, they were also dismissed as junk. The books were written about this, the selfish gene, etc. Now we now know, and I don't know if Jeff's in the audience. Are you here, Jeff? Oh, there you are. Sorry, I saw you. Excuse me. That transposons are really the major drivers of both evolution, and they carry around packets of regulatory information, which is where the system evolves very quickly, and then it has other uses and makes other problems. And the third surprise, which is a bit later in the genome projects, was the numbers of genes doesn't scale with developmental complexity. And articles written in Nature was, oh, well, the same number of transcription factors in C. elegans and humans, but they act combinatorically, and so that's enough. And what was unstated was that we assume that the combinatorics of transcription factor interaction, and if you've got 3,000 transcription factors, 3,000 prime combinatorically is a number bigger than the molecules in the universe. That's more than enough headroom to control worm or human development. That unstated assumption was never justified theoretically, mathematically, nor mechanistically. But everybody just went back to studying their same favourite transcription factor, secure in the knowledge there was enough headroom from transcription factor combinatorics to control human development, and just ignored this stuff, noise. So let's look at this. And it's sort of things that most of us don't think about, in the sense that we're concerned with fragilities, biochemical and physiological fragilities that affect health once we've reached maturity. But we've got to get there in the first place and get 40 trillion cells in the right places. So the worm on the right's got 1,000 cells, a few muscle cells, a few nerve cells in the gut. The organism on the left's got 40 trillion cells, 40,000 million cells. Exquisitely organized, I'll show you in a moment, into a myriad of bones, muscles, and organs. They have the same number of protein coding genes, called the C value and G value enigma. What's worse is most of them, many of them, are orthologous. So the question arises, how much information is required to program our development from the point of conception, from a single cell to this, right? You know, big deal. How much and what form? So it's my favorite organs, by the way, bones, apart from the brain. They're exquisite. Every one of them has a different architecture. So these are 3 vertebrae from different parts of the spine in 3 different planes. You can see how exquisite the architecture is. And an anatomical pathologist can look at a skeleton and tell you exactly which vertebrae is C5 or C3 or whatever. Think of the pelvis, the fingers in the bone. I mean, the bones in your finger, fine bone in your ear, etc. Every one of them has to be architecturally specified. And so it's not just cell type specification. There's not 200 cell types, there's trillions, because it's a spatial organization of these cells. And development, 1 cell, 2 cells, 4 cells, 8 cells, has to make a decision at every stage when to get off the divisional bus and differentiate into an osteoclast or a fibroblast or whatever. Look at the muscles, every one of them. You see this in physio offices. Every one of them is different connections. So we know in C. elegans, because it's been mapped out, the ontogeny of every cell in development has about 1,000 cells, which means it's got 2,000 cell fate decisions. In human ontogeny is equally precise. So if you look at monozygotic twins, they're essentially phenocopies. So the program that manages our development and the differences between us as individuals is not, there's no noise. If there is, it's not relevant. It's really precise. So in the case of worms, that's 2,000 cell fate decision. In the case of us, it's 80 trillion cell fate decisions in a divisional hierarchy. Let's say that number slowly. 80,000, 1,000 million cell fate decisions have to be made with perfect accuracy. And that number is 4 orders of magnitude larger than the maximum information content in the human genome. So, been known for a while, non-coding extent of our so-called junk DNA increases with developmental complexity, topping out humans at 98.8% non-protein coding DNA. And we've known for 20 years, another surprise, another anomaly, not just introns, but irrespective of the extent of these sequences, they're all dynamically transcribed in different cells and tissues at different stages of development. People say, oh, well, it's just cell type specific noise. I mean, how intellectually stultifying is that? Say, I don't understand, it's got to be noise. So you're forced to the conclusion that either there's a tremendous amount of useless transcription or there's another layer of genetic information that's being transacted by RNA, which expands with complexity. Now, the data that came from this, and David Hume's in the audience, David actually organised our participation from IMB in the Recon Phantom Consortium that did high throughput sequencing of cDNAs back in the early 2000s. And the motivation for this study was mainly to try to define the proteome and the spliceosome more precisely. But the ultimate, unexpected, but ultimately headline conclusion was that they picked up tens of thousands of transcripts that had little or no protein coding potential. The accompanying paper we showed, that was just a minor contributor. David was, led a contribution from IMB that the antisense transcriptions everywhere, and it looked like that this was playing some regulatory role. A few months earlier, Tom Gingeris at Cold Spring Harbor, Affymetrix showed the same thing using a different technology, and also showed something that keeps getting overlooked, that almost half of all transcripts are not polyadenylated. And in fact, the major source of non-coding transcripts in the cell, as shown later by Phil Kapranoff, who's on this paper as well, is actually RNAs derived from introns. So they're not rapidly degraded at all. They're actually quite stable and they play roles in the cell. So one quick point I want to make, which is also not appreciated widely, is that there is no such thing as a discrete gene, not that I'm aware of. The genome is a transcriptional continuum. You've got protein coding and non-coding transcripts on both strands that overlap with small RNAs encoded in the introns or elsewhere, etc. And this cartoon, which we drew a few years ago, is really an oversimplification of the actual landscape. Now, I'll give you one quick example. Tom Gingerison, his team showed that in early embryogenesis, in both mammals and in Drosophila, there's an exon that's hundreds of kilobases upstream, which is stuck into the transcripts. That so-called intron is jumping over many other genes in between. So in the adult, the start site's down here, but in the embryo, it's hundreds of kilobases downstream. We don't know why and what that first exon's doing, but it's a continuum. So it's very difficult to disentangle these relationships. So it's an old paper now, but these loci that produce these RNAs have all of the features of real genes. In fact, what Riken showed was that the promoters are actually enigmatically more highly conserved than promoters of protein coding genes. My feeling is because they're more tissue specific. They have a whole bunch of signatures, including all of the chromatin signatures associated with real genes, etc. They're regulated, they show very specific expression patterns. They also have a very similar range of half-lives over orders of magnitude, as do messenger RNAs. So some are very long-lived, some are short-lived, just like protein coding transcripts. I'm moving quickly because I want to get through a fair bit of stuff here. This is a paper that was done by Marcel and Tim in my lab at IMB some years ago, looking at the expression patterns of RNAs. The Riken clones in brain was actually data from the Allen Brain Institute. And if you look at panel B, for example, there's an RNA that's only expressed, as far as we know, in the dentate gyrus. The next one's only expressed in the CA1 subfield of the hippocampus. Others, I should go back, in different layers of the cortex or the cerebellum. Just parenthetically, we've knocked out B, and the mice lose hippocampal memory, but not cortical memory. We knocked out one of the cerebellum ones, and the mice lose spatial navigation, but only in the females, not the males, which is, there's a stereotype to play with there. Sorry. Don't kill me, guys. So what Marcel and Tim showed was that looking at over 1,000 of these things, that most of them were expressed in brain, and most of them showed very cell-specific expression patterns, which, because these things are fairly lowly expressed, they were easy to dismiss as noise. But in fact, they're not lowly expressed. They're just very specifically expressed. You can see the in situ hybridization patterns as clear as crystal, but only in a few cells. So you mash up that mouse brain, you'd be lucky to see these transcripts because it's only in a few cells. Many transcriptome studies are just the sequencing of smoothies, tissue smoothies. They also show very specific subcellular localization. Surprising number go to the cytoplasm. I guess roughly about 30%. And by the way, I will allude to this quickly later, but the cytoplasm is organized exquisitely into phase-separated domains that are nucleated by these RNAs. Even metabolic pathways, translation, all takes place in phase-separated domains that are formed by the interaction of RNAs and proteins. Most of them are in the nucleus and chromatin associated. We'll get to that. Some decorate unknown organelles, subnuclear organelles, in this case, Purkinje cells, paraspeckles, and or trafficked down synapses. And Tim Brady over at UQ is doing a fantastic job in tracking link RNAs that are being moved out of the synapse and modified in response to neuronal activity. So one of them is a thing called GAMAFU, which actually decorates modified spliceosomes, and we were able to show that this one is acutely downregulated in response to neuronal activity and changes the alternative splice patterns in the same way that you see in schizophrenia, and associates with, I'm not sure if I've highlighted it, yes, see, splicing factors, quaking-1, SRSF1 in the spliceosome. So here's a long non-coding RNA that's modifying splicing responses to neuronal activity in a subset of neurons. So Tim went off to the Broad to work with John Rinn to develop this RNA equivalent of exome sequencing, where you capture the transcripts from a genomic region you're interested in, and you can deep sequence it. Because we knew that regular transcriptome stuff of brains or kidneys or anything else was sequencing smoothies, and we weren't seeing what's really happening at the cellular level. So this is like a molecular microscope. So that's the technology, you sequence it. So the first one that Tim did was look at fibroblasts in culture. This circos plot here represents regions of the genome that did not appear to be expressed in classic lemma short read sequencing of the total RNA from these fibroblasts in culture. So if you look at the one, the segment on the lower left, around 7 or 8 o'clock, there's nothing we could see. So Tim developed hybridization probes to capture any transcripts that might have come from these regions, hybridized them, and then sequenced, and from the same prep that we used to generate this data. And this is what he found. These things are alive with transcripts. They're alive. And every one of these arches is a splice junction. So there's obviously a subpopulation of cells, even in fibroblast culture, which are in different transcriptional state. So these aren't gene deserts at all. They're just too small a fraction of the population to be picked up by bulk RNA sequencing. We did the same thing with classic protein coding regions like Hox genes, P53, etc. Found all these new non-coding RNAs, as well as new isoforms of even things like P53, which have been intensively studied. So keep in mind that averages mislead. A lot of so-called gene network studies say, oh, this one's going up, that one's going up. They're somehow connected, but they could be in totally different cells, types of cells in your culture. So I think we should take more notice of at the time, and to move quickly, is that the extraordinary complexity of the alternative splicing of these link RNAs. So pre-capture, in that experiment that I just talked about, we got 3 exons of this thing, but when we captured it, we got all of these exons. Look at them, and look at all the splice junctions. There's only one internal one, I think, that's not alternatively spliced. Incredible. Now, the splicing pattern isn't random, but every exon is alternatively spliced to more than one other. So we didn't take enough notice of this, because what was in literature, because of slow depth of sequencing, was, well, many of these things were 1 exon or 2 exons or whatever. But it was really because the depth of sequencing wasn't enough to produce accurate transcript isoforms. So Tim, with very great student Iris de Garvin, did capture seq the entirety of chromosome 21 and the mouse syntenic regions. At the time, sorry, we'll go back. That was the current annotations in GeneCode at the time. They got so much depth of data that this is what it looks like after they'd finished sequencing. What they discovered was that across the whole chromosome, that the 96% of all the exons that they picked up in link RNAs were alternatively spliced. Now, this means they're highly modular, and I'll talk about plug and play in epigenetic regulation shortly, but much more so than messenger RNA exons. Now, interestingly, that 69% messenger RNA is an exaggeration, because most of the alternative splicing is actually in the non-coding exons at the 5' end. So the well understood increase in alternative splicing between humans compared to mice is actually not more alternative splicing in protein coding sequences. It's actually more alternative splicing of the regulatory exons, the 5' end. So we got the same result in mice, mainly alternative splicing, and these are real splice junctions. So if you look at the coding exons and the patterns, genome or chromosome wide, you can see the patterns for coding exons. The non-coding exons are virtually identical. Subtlety here is the ones, the non-coding are actually slight subtlety differences are associated with alternative splicing. And we know that from protein coding exons are alternatively spliced as well. So these are real splice junctions that are evolutionarily conserved. This ain't noise. If these weren't relevant, these would have degraded eons ago. These are real transcripts that are regulated expression and have regulated splicing patterns. So that's the summary, but basically, and I left the slide out, but if you look at the top left there in A, the blue exons towards the right of that figure are the protein coding sequences, which are constitutively spliced, which is true in most genes except multifunctional larger proteins. All of the alternative splicing in the human there at the left hand, the 5' end, is non-coding exons. In mice, you see no alternative splicing in that area. And we think that's got to do with cognitive capacity. In fact, that gene is one of the genes in the trisomy specific region, chromosome 21, that affects mental processes. So I want to just, and I think this can be useful to you. We've now shown that most regions associated with human complex traits and diseases are expressing long non-coding RNAs. Now, you'll be familiar with GWAS studies, and Manhattan plots, so I won't explain that to you, but this is one of my favorites. And in fact, we're now identifying the rodent orthologs of the link RNAs coming out of all of the haplotype blocks associated with human intelligence and poking them with antisense oligonucleotides and touchscreen computer analysis of their cognitive abilities using techniques developed in Cambridge. But I digress slightly. But the opportunity here is actually to get to molecular etiology of many of these traits. So you know that most of the haplotype blocks that are associated with complex traits and disorders do not encode proteins. The variants are in intronic, intergetic, and other non-coding regions. Only a small proportion are in coding sequences. So this is different to Mendelian disorders like cystic fibrosis or thalassemia, which are catastrophic component damage. This is regulatory variation, much more subtle. In fact, that's why the genetics is more subtle, because unless you make deletions in these sequences, they're still functional. The differences between us is due to variations in these sequences, some of which give us fragilities that lead us into unhealthy spaces. So Marcel and Nenad back at Garvin did a capture-seq for all of the 150 GWAS regions that covered at that time 150 different conditions. So made hybridization probes, took RNA and sequenced it, and this covered about 2% of the genome. So the depth wasn't huge, but it was still 50 times greater than just doing the whole thing. And what they found was that most of the GWAS blocks expressed non-coding RNA. So here's a good one, another one to do with intelligence and cognitive performance. The red bar at the top left there is the haplotype block. That's the region that it covers in the genome. If you look down the bottom, at that time, there was nothing recorded in GenCode. The sentinel SNP that's used in the arrays is there. When we sequenced it deeply, we found a multi-exonic, alternatively spliced link RNA sitting right in the middle of this haplotype block, which has to be a prime candidate for the molecular etiology of whatever is involved in the differences in cognitive performance in the population studied. And in fact, we got most GWAS loci expressing long non-coding RNAs. Here's another one that's got 3 of them. And I got this quote from a Nature Genetics review recently. Although most causal genetic variants for Mendelian disorders fall in protein coding regions, we know that, the heritable component of common disease risk distributes largely to non-coding regions and appears to be particularly enriched in enhancers that are specific to disease-relevant cell types. Now, I'm going to fast forward a bit and tell you most of these long non-coding RNAs are the products of genes called enhancers. And that really gives you the conceptual framework for understanding what's in the human genome. So we did the same thing Simon, who's now with BioNTech in Melbourne, did the same thing for haplotype blocks associated with neuropsychiatric functions. I don't expect you to read this, I'm sorry, but we picked up majority of the haplotype blocks associated with neuropsychiatric disorders transcribed into multi-exonic link RNAs, most of which were not recorded in the database. In fact, we're now knocking these down in rats. We've got the rodent orthologs. By the way, there's more rodent orthologs of the link RNAs from haplotype blocks associated with neuropsychiatric traits than there are with intelligence, which makes sense because we're a lot smarter than rats, although they're pretty smart. But so these are actually fragilities and more basal pathways that give you neuronal dysfunction. And what we'd like to do is do what Juliet French in her lab, Vina's in the audience. Is Juliet? Where is she? She's not here. Oh, there she is. Oh, that's mine. Sorry. This is great work from QIMR. So they are now validating, and I'm actually, you probably see my suggestions for the abstract minor. So this, to my knowledge, is the first paper that's actually linked at a mechanistic level, a link RNA coming from a haplotype block associated with a risk. This is breast cancer risk in this case. And what Juliet Minor, name's up there, of course, has shown is that the long non-coding RNA, they've cleverly called killer, is responsible for the mechanistic effect of the lockers in those people shown by GWAS stuff to be susceptible to breast cancer. And down to mechanism affecting, I think, from memory DNA repair pathways. And they've got another one, beautiful one, in about to go out. So I think a good clue for all of us interested in understanding the molecular basis and therefore the first step towards therapeutic intervention of complex traits and disorders, whether it's type 2 diabetes, neuropsychiatric disorders, any other, is to go looking quickly for the link RNA that's in that haplotype block. You've got the genomic data, see if there's variants that distinguish the so-called normal versus the affected population. And you've probably, I think, got almost certain chance of it working out what is the molecular basis of these problems. So early on when we were doing this, and David will remember this, you know, people will say, oh, it's just junk. So we had to show that these things were differentially expressed in different contexts and poke them a bit and see what happened, you know. Like one swallow doesn't make a summer. So you've got to sort of step back and say, look, and so we looked at the differential expression of these things in a whole bunch of processes: embryonic stem cell differentiation, neuronal stem cell differentiation, T cell activation. That's an interesting one. Muscle differentiation, cancer and memory development, and in melanoma. So I was going to show you two very quick examples to give you some tangibility on these things. So first one's from memory development. And those days we used microarrays, and which we made ourselves and then commercialized through Invitrogen. And we found almost 100 RNAs were differentially expressed in breast development. And we focused on this one, which was the second top hit. It was antisense, nearby antisense, not overlapping, zinc finger transcription factor gene. And this one is very highly expressed in pregnant mammary gland, then turned off when the mice are lactating, feeding the pups. And when they're weaned, this RNA goes back up again. So keep this in mind. This one is down in lactation. So we looked at it. It had a very long half-life. This is actinomycin D dunked down. The blue line is the long non-coding RNA. The red is the nearby protein coding gene. It's bulletproof. It's probably resistant because of chromatin associated. And this is done in mammary epithelial cells, I should say. So when we knock it down, the cells proliferate more and form some dome structures, which is formed in these epithelial cells at confluence. But on the far right there, when we knock this RNA down, remember it's down in lactation, they start producing casein, the major milk protein. These are mammary epithelial cells in culture. These are primed to differentiate into producing milk proteins. And all we had to do was flick the switch by turning off this RNA. We looked at another one, which is, another one that was involved in melanoma, commonly expressed in melanoma cells. This is the one we picked on. It's a small RNA coming out of the intron of this other gene. And it was small enough to get pretty accurate secondary structure predictions. And I put this up just to illustrate it with a point. RNAs are not wiggly lines with A's on the end. They're extremely complex three-dimensional structures. They can form hydrogen bonds on all three faces, including the 2' hydroxyl, which is what distinguishes RNA from DNA at a structural level. So here you've got very well-structured secondary structure stem loops that bind proteins, and you've got open regions that can interact with other RNAs or DNA in a sequence-specific manner. So these things can act, and that's what they do, as scaffolds and guides. The scaffolds are the secondary structures that bind proteins. The guides are the sequences that can hybridize to target sequences in a chromatin. I'll show you. So if we knock this one down, we can knock down the invasive. This is a metastasis gene. And if it's small enough to ectopically produce, and this video doesn't work for some reason, but basically just to explain the result we got, if we do a scratch test and look at motility on the left versus overexpressing this small RNA on the right, which is up in melanoma, those cells will migrate much more rapidly. They're much more motile, much more invasive. I've just got to step through what doesn't work. So zillions of publications now showing, there's a million stories about different RNAs having different roles in normal development, different pathogenic processes. So, and they show all sorts of different phenotypes. A recent review we wrote for NSMCB. When you delete them, you see drastic phenotypes. But most single nucleotide variations don't show anything other than a phenotypic variation. It's not catastrophic. And there's now many cases of them being shown to be involved in cell fate programming and reprogramming. So Marcel and Paolo looked at dynamic expression of these things in embryonal stem cell differentiation. And I think I'm going to run out of time unless I keep moving. What they found was that 2 of these long non-coding RNAs were highly differentially expressed, and that they are associated with trithorax, MLL1, which is the protein that's commonly fused in leukemias, and with activated chromatin, with histones that are trimethylated on lysine 4, which was the first demonstration that linked RNAs are associated with chromatin-modifying proteins, activating proteins, I should say. At the same time, elsewhere, other groups have shown that link RNAs were associated with chromatin repressive proteins like polycomb. G9A is just a short form for polycomb. And basically then John Rinn, a year later, doing what only the Broad can do, looked at 3,300 in parallel and found that 20% of them actually were physically associated with polycomb chromatin modifying complex, which modifies histones epigenetically, and others were associated with other chromatin modifying complexes. So we know that epigenetic processes, central differentiation development, long-term responses to environmental variables, and brain function. You knock out epigenetic processes, you can't learn. So the epigenetic information, the memory is embedded not just in the methylation of cytosines, which is overemphasized because they're easy to study, but really in an extraordinarily wide range of modifications of histones that package DNA nucleosomes. And by the way, these modifications include dopamine and serotonin in the brain. So your neurotransmitters are also being physically covalently linked to the histones. So your neurotransmitter activity is also being laid down as an epigenetic memory in the brain. And think about the implications of that for experience affecting behavior or in memory. So there's a lot of enzymes involved. They have no sequence specificity. They're thought to be guided by transcription factors. No evidence for that. So the question is, what determines the site selectivity of these enzymes? Because they do differentially modify histones at many different sites in the tails, at zillions of different places around the genome. So what determines their site specificity or selectivity? What determines the positioning of nucleosomes? I don't have time to talk about it, but we and others show the nucleosomes are specifically located and specifically over exons. So the genome knows where the exons are, not just in messenger RNAs, but non-coding RNAs. I can't go into that today. And what's the molecular basis of environmental epigenome interactions? And the answer to all three of these questions is RNA. So papers over the last decade and a half have shown there's widespread transcription and neuronal activity in regulated enhancers, that long non-coding RNAs have enhancer-like function in human cells, and that there is now direct evidence that the mechanism of enhancer action is mediated by long non-coding RNAs by CRISPR-Cas13 shRNA knockdown, and studies showing that alternative splicing is changing the activity of these things. So basically, most link RNAs, maybe 60 or 70%, are coming from genes called enhancers. And there's other papers that reinforce this in different ways. So even the globin lockers with so-called locus control regions that cause chromatin looping and differential expression of the hemoglobin genes, is preceded by and requires it's enhancer RNA transcription. So enhancers, for those of you who aren't familiar with them, have been known for a century, although only called this perhaps in the 80s. They've been known in Drosophila, bithorax, and other genetic loci. They're genetic loci, they're called, that control the spatio-temporal patterns of development. Now back to 1 cell to 40 trillion. So these are the parts of the genome that are controlling the spatio-temporal patterns of development, all those cell fate, divider, differentiation choices. There's estimated to be, very roughly, about 1 million of these things in the human genome. So there's not 20,000 genes in the human genome, there's 1 million. But most of them do not encode proteins, they encode enhancer RNAs. Now that's a very rough estimate, but it's ballpark. The mechanism of enhancer action has long been a matter of speculation, and a popular model put forward in the 80s, now it's the 1980s, nobody knew anything. But the transcription factor gurus, particularly one guy called Mark Potashny, who basically said that epigenetics is rubbish because the chromatin modifying proteins didn't have site specificity, so everything was being controlled by transcription factors. So he puts forward this method. They start to realize there are other genetic loci, both in Drosophila and mammals, that control spatio-temporal patterns of development, and they don't encode proteins. So here's his model. Because everyone knew, and this is true, that enhancer action involves reorganization of the chromatin, so-called chromatin looping, into topologically associated domains that are transcription splicing hubs. So he says, well, the enhancer binds transcription factors that then loop around to contact the promoters of target genes in some sort of club sandwich. See the club sandwich there, protein-protein interactions? Now, how specific could that be? I mean, but everyone believes this, and I still go to talks where people talk about enhancers and put this model up. There's no evidence for it, and it's absurd that you could have a protein that somehow feels around for another protein, likes to stick to, that's somehow bound to another protein that's bound to an enhanced transcription factor, the promoter of some other gene. And it gets worse, because you have one enhancer targeting many genes, and you have one gene targeted by many enhancers. There's like intellectual consortiums, but people believe this. It's incredible how preconceptions become fossilized. So what I've observed in the history of science is that when people make an observation, they legitimately, correctly try to generalize it. So if the truth of that generalization can't be tested at the moment, it becomes the accepted explanation, then the orthodoxy, and then an article of faith. And I have zero evidence apart from a speculation. This one in case 1986. Amazing. So enhancers result in chromosome looping. You can read that fast as I can say. This actually involves, and this is really important, as the frontier of cell and developmental biology, as well as neurobiology, is the formation of phase-separated biomolecular condensates, both in the chromatin, but also in the cytoplasm and at the synapse, for that matter. So what's the mechanism? Is it protein-protein or RNA-protein, RNA-DNA interactions? So we know that transcription correlates with enhancer action. It requires the assembly of an enhancer RNA RNP condensate, that important proteins in enhancer action, topologically associated domain formation in the nucleus, are recruited to their positions by RNAs, and that the RNA has been shown in a number of cases to modulate DNA and histone modifications. So link RNAs associated with the complex, sorry about the text, but I just got to get through this quickly. If you knock them down directly by shRNA or Cas13, enhancer action is abrogated. So the transcription of link RNA is not a byproduct of transcription factor binding and enhancers. It is the product of that gene that then mediates the formation of the topologically associated domain and enhancer action. You knock out the RNA directly, you lose an enhancer. It's modulated by splicing, as I said. RNA modifications are involved, but I have to skip over that. And we now know with many examples that these things activate. Yeah, I see the time. I'll have to move quickly. By binding to DNA through triplex formation. And the high-resolution imaging shows the localization of these RNAs in phase-separated condensates in the nucleus. So here's a study of many of them showing a look at the spots. They're all localized in punctate domains. I have to move quickly. One of my favorite slides, this is interphase fibroblast nuclei with anti-triplex antibodies. And the thing lights up like the Milky Way. There's RNA DNA triplexes all over the genome. And this is base pair specific in the so-called Hookstein groove of the DNA. So they're genes. Enhancers are genes. And I'll just flick through this, but it explains the production of most link RNAs from genes called enhancers, explains their low cell-specific expression, their mostly nuclear localization, their rapid evolution under positive selection for adaptive radiation, and also the genome-wide instance of the features of regulated gene expression, transcription start sites, splice sites, etc., documented by the ENCODE projects. So it also resolves the G-value enigma because we got a lot more of these things than worms. And the high degree of alternative splicing in these RNAs enables enormous flexibility and modularity of protein binding and target selection, which in conjunction with the post-transcriptional modifications of the intrinsically disordered regions in the proteins with which they interact, provides a plausible mechanistic framework for directing the trillions of cell fate decisions that must be made accurately during human development. So I'm going to skip over this, but I just want to make a couple of quick points that you probably should be aware of this. This is the biggest advance in cell biology in the last decade, is the realization that most of the cells are organized into phase-separated domains. And they're formed by the interaction of RNAs, guide RNAs, with the intrinsically disordered regions of proteins, which is where nearly all the post-translational modifications occur, like in the histone tails. They're essential, they're in and essential for the function of virtually all of the proteins involved in development, including RNA polymerase, so translation takes place in a phase-separated domain. So does transcription. I have to move quickly. And the fraction of proteins containing these correlates with developmental complexity. It's the only part of the proteome that does. And we know that the chromosomes are organized into phase-separated domains called TADs. So this is a new model. It's pretty crude. I actually even said in the paper, one should not believe these cartoons because they can be misleading. But what this is trying to say is that a chromosome looping topologically associated domain is nucleated by the RNA that brings then the proteins into that domain and targets the relevant areas of DNA by DNA binding to form these condensates, and that this is the critical step in making cell fate decisions during differentiation development, not just in development, in the immune system and in the brain. So they're very modular. I'll skip over that. We know these motifs abound in the human genome, and you can map them all around the genome. So you'll see polycomb binding domains everywhere, etc. And I think there's going to be a new RFAM like PFAM. So, in PFAM, you can say that's a serine protease domain or a kinase domain. We'll be able to say, and we're working on this, that's a polycomb binding domain, that's a trithorax binding domain, etc. And so we already know some of these domains as validated that they're binding specific sorts of proteins. We also know that, and there's something to keep in mind, that RNA is also the substrate for environmental epigenome interactions through RNA modifications and RNA editing, which we're just starting to study through direct sequencing with nanopore. This is work with Eva, postdoc in my lab's now got her own group in Barcelona. Fabulous stuff that she's doing to try and understand, and Tim Brady's doing this with synapses over at UQ, the RNA modifications that are being made in response to synaptic activity or other processes. So, to finish, in 1980, nobody knew what was in the human genome, except that a lot of it seemed to be junk with introns and or repetitive sequences. Nobody knew how many protein coding genes there were. They speculated maybe 100,000. Who knew? By 2000 with the genome projects, we knew there were only 20,000 protein coding genes, picked up a bunch of other interesting RNAs, but the rest of it was either introns or repetitive sequences, assumed to be non-functional. I think this is much more accurate now, that a small fraction of our genome encodes the Lego parts. The muscle proteins in us are the same as they are in worms. Most of it is the programming of a development. You think about a Lego set, the set of instructions to build a house or a castle, the instructions to build the castle are much larger and using mostly the same components, although there's a few new ones. So the genome is not, a desert of junk with occasional oases of protein coding sequences. It's actually islands of protein coding sequences in a sea of regulation, most of which is transacted by the RNAs. And those RNAs are primarily involved in organizing the cell and the chromatin for proper execution of differentiation development. Most of the work was actually done over at IMB. And they're the people that contributed to the stuff I've talked about today, then at the Garvan and our collaborators at Recon and other places. Thank you.